Everyone. In this lab, you're going to learn about different water testing mechanisms that are actually used by companies today. And these different water testing procedures allow us to see if water is drinkable. So we test for certain bacteria, which I will talk about, and we do these group of tests. When I say positive, it means the bacteria are present. So the first test in water testing is called the presumptive test, which you would have done in lab. If that's positive and bacteria are present, then we do the confirm test. And if that's positive and bacteria are present in that water sample, we do the completed test, and you tell the whatever water district you're working in that your water is either contaminated with this type of bacteria or not. So we will talk about all of this. So I'm going to start off with a little bit of background about water. Water, drinkable water in science is called potable. So anytime you hear the word potable, it means drinkable. If I tell you water is not potable, it means that it's not drinkable. It's probably contaminated with microbes. And here we're focusing on bacteria. So water can be contaminated by many different things, but typically it's contaminated by human and animal waste. And so that's why hygiene and sanitation is very important. And that's also the reason why a lot of countries have a lot of diseases that you may not see in developed countries, because hygiene and sanitation is so big. So if you live in a certain area and you have farms and you have animal runoff coming in or sewage, this can really be a devastating source of disease for that community. So we can see fecal bacteria, meaning bacteria from people's feces or waste or animal waste, parasites, viruses, and all of these can be passed down to the host, the human host, through either drinking that water, using that water to take a shower, laundry, any of those things. So it's very important to test water, and microbiologists are typically the ones who test water. And according to the Center for Disease Control, poor water supplies and sanitation account for almost 90% of death from diarrhea. So it is a major world health problem, these different water diseases, and you guys are going to learn about the water testing that's done to determine if that water is potable or not. So water testing is done routinely, and it would be very hard. So if, for example, if you have a water source and you collect a sample of water, it's too expensive and too much work to look for every possible contaminant. So you cannot possibly look for every single pathogen in that water. It would be too much work. It wouldn't even be possible to look for every single pathogen, because we're not even aware of all the pathogens. And pathogens can enter the water at any time. They can be very difficult to culture in lab, even if we had the mechanisms. And most importantly, once a pathogen is detected in water, it's usually causing an outbreak. So we want to try to test the water continuously before pathogens are there. And again, we cannot possibly test for every pathogen. So what scientists do is they look for these organisms called indicator organisms. And the indicator organisms that we use in water testing are called coliforms. Coliforms are normal bacterial flora that are found in the intestines of humans and animals. Right now, if you were to culture the intestines of myself or animals like cows or a different farm, if you were to take a sample, we would see coliforms, which again are just the normal bacteria that are present in the intestines. Now that bacteria gets excreted in feces, so that's why we know that if we see those bacteria in a water source, it means that we have waste runoff either from humans or animals, and this can make people very sick, and it can be indicative of a lot of different contamination of that water. So coliforms, they're indicator organisms, and the reason we use them is that they're very easy to grow in lab or culture in lab. That's what culturing in lab means. And they indicate the potential contamination of water with fecal pathogens. So coliforms, the exact definition of them that I want everyone to leave class knowing is they're facultative anaerobic gram-negative rods which produce acid and gas from lactose fermentation when incubated at 35 degrees Celsius for 48 hours. So I want everyone to know this definition of what coliforms are, and let's go through it. Let's break it down a little bit. So coliforms are facultative organisms. Facultative means they can grow anywhere. They can grow with and without oxygen. They're, that's facultative anaerobic. They're gram-negative rods. So you guys know what gram-negative is. They have an outer membrane and they have thin peptidoglycan. They're rod-shaped. So when you grow these organisms in lactose broth, which is a sugar found in milk, for example, they will use that sugar and they will produce acid and gas as a byproduct of their metabolism when they're incubated for 2 days at 35 degrees Celsius. So a little bit less than body temperature. So again, I want everyone to know that coliforms are facultative anaerobic gram-negative rods which produce acid and gas when they ferment lactose at 35 degrees Celsius for 48 hours. So with water testing, what we will do in lab is we're going to follow a protocol established by the American Public Health Association. And again, this goes through the 3 tests I talked about. First, you do a presumptive test in lab where you inoculate different dilutions of your water sample into lactose broth, and you see if the bacteria produced acid or gas. If that sample is positive, you don't go right away and tell the city or the county that the water is contaminated. You do another test to confirm, and that's called the confirm test. And that confirm test uses this EMB agar place, which we will talk about, to look at coliforms and determine the type of coliforms. And one reason we do the confirm test is because the presumptive test can give you a false positive. And then finally, if that gives you a positive result, meaning coliforms are present in your water sample, you do a completed test, which again, just does more gram staining and more growing the organisms in lactose, just to get better idea of what exact coliforms and potential pathogens there are. So we're going to talk about the presumptive test. So the presumptive test is always the first test that's done with water testing. So we take a water sample, and in lab, what we typically ask students to do, which we're not doing right now with the online world, is I ask students to bring in water samples, and sometimes people will test the community college swimming pool or their own swimming pool or the animal, like if they have a dog, the water place that the dog drinks from, just to see, is it contaminated? Because I know some people never clean that at all. So let's imagine we have your water source. Let's imagine a swimming pool, and you're trying to determine, is it contaminated with indicator organisms, and we're using coliforms. So the presumptive test, you do is you take different dilutions of your water sample, meaning you add different amounts into lactose broth. This is what lactose broth looks like. It's green, and it has a pH indicator, which is blue. I mean, you can say blue, green, whatever. This is what it's starting out with. And in this lactose broth, there's a little tube, which you can maybe see in these. It's a little tube inside the big test tubes, and that is called a Durham tube. And the reason we have that is because if bacteria produce gas, we can see a little bubble. So that's why we have the Durham tubes. And so you have your lactose broth, you do different dilutions of your water sample, you incubate those tubes that have your water sample at 35 degrees Celsius, because coliforms, if they're present, they grow in 48 hours at 35 degrees Celsius, and then you go back and see the results. If coliforms are present, they can ferment or use the lactose, and they produce acid and gas as a response. The way we see acid is the lactose broth turns yellow because the pH is acidic, and we see gas production. We see a little bubble in the Durham tube. So we test acid production by a change in medium color. Yellow means that they did produce acid, and then you test a gas production by looking at the Durham tube. If you see a little bubble, that means that they did produce gas. So for there to be coliforms, you need to see both acid and gas, meaning that when you look at your tubes, they have to be 2 things. They have to be yellow, and you have to see a bubble in the Durham tube. If you just see yellow, it doesn't mean that they're positive for coliforms, because coliforms have to do those 2 things. They have to produce acid and gas. So that's how the presumptive test works. And again, here's the definition of coliforms. They're facultative anaerobic gram-negative rods which produce acid and gas. So that's the key, from lactose fermentation in 48 hours at 35 degrees Celsius. And so the way, once you do this test and you pull your tubes out of the incubator, you're going to determine something called the MPN, which stands for the most probable number. It's a statistical estimation of coliforms per 100 milliliters of water sample. And basically what you do is you go through your tubes and you mark, are they positive or not? When I say positive, I mean that you see acid and gas, meaning they're yellow and gas. And you use a table called the most probable number table, and you determine the MPN number. And if your MPN is anything above 0, it means that that water is not potable because there's coliform bacteria. So if you are a microbiologist working for a water company, and this is a great job if you end up doing this job, if you determine that the MPN of a water source, like a swimming pool or anything else, the MPN is above 0, you tell that community that that water has coliforms in it, so it's not drinkable. Now, the presumptive test, which we just talked about, it's not a definitive final test for coliforms because sometimes you could have synergism. The definition of synergism is that you have multiple bacteria in your tube and they're giving you mixed results. So maybe you have two bacteria in your tube. One is producing acid and one is producing gas, but it's not a coliform because a coliform does both. It's one bacteria that produces acid and gas. So we're scared of synergism. That's why we have to do another testing. So if you get your MPN above the 0, you're going to go on and do a confirmed test. So the confirmed test, what it does is you take your tubes that were positive from the presumptive test, that were yellow and had a bubble for acid and gas, and you streak them on specific agar plates called eosin methylene blue agar plates, which we can just call EMB plates. And we try to visualize what the bacteria looks like. Here's an EMB plate, so you just streak your bacteria on there. And the reason we have to do a confirmed test again is because these might be giving you false positives, the presumptive test, because of synergism, because of having two organisms, one that's producing acid and one that's producing gas. And there you're getting a result that looks like you have a coliform, but in reality you have mixed bacteria producing different things. And then if your confirmed test is positive, in water testing, they also do a completed test, which looks at the water even further, doing more gram staining and a little bit more testing to just confirm that that water is indeed contaminated. Okay, so with the presumptive test, here's an example of what results would look like. So again, we would inoculate water samples in lactose fermentation broth. The lactose fermentation broth is green to begin with, and then you incubate. So after you have your water samples, you incubate those tubes at 35 degrees Celsius. So you put them in a little incubator oven for 2 days. Then you pull out your tubes and you look at them. Now you're going to determine what they are using these keys. If you see a yellow color, you're going to say the tube, you're going to label it as A for acid. If you see a yellow color and gas in Durham tube, it's AG. If you see no change in color, that's just a green color. And if they became more basic or alkaline, they turn blue, so you put K for them. So this is what these symbols stand for. And again, to be positive in the presumptive test, they have to be AG. We have to see acid and gas. They have to be yellow and have a gas in the Durham tube. So if I were to look at these tubes, so this is the starting point. This is what lactose broth looks like before you add your water to it. Then you add your water to it and you could get many different results. So let's say these are 3 different water samples. These all turned blue, so the symbol for them is K. They turned alkaline, so there's no presence of coliforms. These tubes all turned yellow, and you can't see them, but if you zoom in, some of these have bubbles, like this one has a bubble and this one has a bubble. So I'd classify these as AG, or positive for acid and gas. And then here's a mixture of tubes. So you just go through these. And then once you have your tubes, you're going to determine MPN. And the way you do this is you go through this table. So when we do the presumptive test tubes, we inoculate 5 tubes with 10 milliliters of water. So we go through. So when I look at these 5 big tubes, not these small ones, and I'm determining what's positive, to be positive, you have to have acid and gas. So you have to have yellow and a bubble. This is yellow and it has a bubble. I can tell when I look really close. This is yellow and it has a bubble. So I have 1, 2 positive. This is not positive. This is yellow and it has a bubble, and this one's not showing, but it's yellow and it has a bubble. So I have 4. So then I go and I circle the 4 right here. So out of 5 tubes with 10 milliliter water, how many are positive? 4. Now it says, look at the 1 tube with 1 ml water. So one of these tubes is labeled 1 ml, and one of the tubes is not labeled, is labeled 0.1 ml. So when I look at this tube right here, it's green, so it's not positive. So I would go to 0 here. Then when I look at the 0.1 ml tube, it is positive. So I would go to 1. So 4, 0, 1. It's easier when you actually circle them. So the MPN for my water sample is 20 here, and this water is not potable. So what I want you to know for the presumptive test is I want everyone to be able to define what coliforms are. I want that big definition for you guys to know that they're facultative, anaerobic, gram-negative rods, that when they ferment lactose, they produce acid and gas at 35 degrees Celsius when they're fermented for 48 hours. This is the 3 steps of water testing. So we went through that. So the first step is presumptive testing, presumptive test, then the confirmed test, then the completed test, interpret what the results look like, know what they look like, know if I give you a picture, which tubes are positive and which not, and what does the MPN have to be for water to be potable? Why are coliforms used in water testing if they are not pathogenic? We talked about that. And why are pathogens not tested for directly in water samples? So I explained that in the beginning. And then explain why the confirmed test must be ran after the presumptive test. So I'm going to talk about the confirmed test now. So now with these tubes, if this was a real water source, I would, as a microbiologist, go on to do the confirmed test. The confirmed test is done on positive, uncertain presumptive test tubes to confirm the presence of coliform bacteria, because remember that with a presumptive test we could get false positives by having synergism. So what we do is we take the tubes that are positive here, so the ones that are yellow and have a bubble, and we do the confirmed test with them. So we're going to talk about the confirmed test, but I want to give a little bit of background. So non-coliforms are non-lactose fermenters, but they could still be fecal bacteria. So there are fecal bacteria, meaning bacteria in your feces, that are not, they don't ferment lactose, they ferment other things. So just know that when they grow on EMB agar, they're colorless colonies, but they're still fecal bacteria. So not all fecal bacteria are coliforms, but a majority are. Coliforms, there's the E. coli group and there's the Enterobacter group. The E. coli group of bacteria is bacteria that's E. coli and a lot of other bacteria that when they ferment lactose, they produce mixed acids. And these ones on the EMB agar, they form colonies that produce many different organic acids. So when you look at the plate, and I'll show a picture in a little bit, because of so much acid production, the colonies turn into this very dark greenish color, so it has a green metallic sheen. So keep that in mind. And then Enterobacter aerogenes bacteria, which are also fecal bacteria. This includes Enterobacter aerogenes and other fermenters of different organisms, different chemicals. So these ones form colonies that ferment lactose and they form colonies, but they produce a little bit less acids. So we get pink purple colonies. So again, when you look at the EMB agar plate, which is just an agar plate, I'll show you, we can get 3 different things. We can get colorless colonies, which indicate non-coliforms. We can get green colonies, which indicate the E. coli group, and we can get purple colonies, which indicate the Enterobacter aerogenes bacterial group. And these are both coliforms. Okay, so the procedure of this is you're going to culture bacteria from positive presumptive test tubes, which are yellow and have a bubble, onto this eosin methylene blue agar plate. So it looks like, to me, it just looks like a red agar plate, and it looks reddish purple. And this agar has lactose and it has dyes. It has red and blue dye. So red and blue make purple. So the plate looks, to me, it looks reddish, but a lot of people say it's purplish. And the EMB agar in science, we say it's a differential medium. Medium just means what you grow bacteria on. It's a differential medium because it allows us to differentiate coliforms from non-coliforms by dyeing coliform bacteria purple, and it's a selective medium because only gram-negative bacteria can grow on it, because there's a lot of dyes that prevent gram-positive bacteria. So here are results from EMB plates. So here we see colorless colonies. So these ones are non-coliforms. Here we see green colonies, so it's a differential medium because we can differentiate coliforms from non-coliforms. This is the E. coli group of coliforms. These are pink colonies. This is the enterobacterogenous group of coliform bacteria. And then on this plate, we see mixed results. We see a little bit of green, a little bit of pink, and colorless. So this water sample is mixed. It has coliforms on it from 2 different groups. This is a pure plate, and this is all 3 of these are pure plates because we just see one group of bacteria on them. So you're going to use the streak plate technique, and you're going to streak your bacteria on EMB plates. So you're going to streak it from your positive presumptive plates, and this is a confirmed test. And you're going to incubate for 48 hours at 35 degrees Celsius, and then you get the results. So the main thing we want you guys to know is to identify the 2 typical coliform groups and differentiate them from non-coliforms. Remember that on the EMB plate, we can have 3 things grow. We can have non-coliforms grow, and they produce clear colonies. So this is non-coliforms, and we can have coliforms grow. Coliforms can either be the E. coli group, which produce green colonies because of all the acids that they produce as part of their metabolism. And then we can get enterobacterogenous, which are also coliform groups, and they produce pink/purple colonies. So non-coliforms, again, they are still fecal bacteria. They're just not coliforms because they don't ferment lactose. And then we talked about the coliforms. So go back and read all of these. And then here we have a mixed plate that has it. I can see it has the E. coli coliform group on it, has a little bit of the pink Enterobacter coliform, and I see some clear colonies which are non-coliforms but still could be indicative of fecal bacteria. Okay, so for the confirmed test, what I want everyone to know is, why is a confirmed test needed if the presumptive test is positive? You can't, after just doing the presumptive test, if you work for a water company, go tell that water source that it's contaminated. You have to go on and do the confirmed test with EMB plates, because your presumptive test could be producing false positives because of synergism. Know the name of the differential and selective medium for coliforms. So that was the EMB agar, which we talked about. It's a differential medium because it differentiates coliforms from non-coliforms, and it's a selective medium because it selects for just gram-negative bacteria to grow. And be able to read what these plates mean, know a pure culture from mixed culture, and then I know the different typical types of coliforms, like the E. coli group and the purple Enterobacter group. So go through all of these and take your time reading this lab and going through it.